spread? Is it an epidemic or is it not an epidemic? If it spreads, how many people will be infected? And therefore, how many people of that fraction who are infected, how many people do I expect will need to be hospitalized? How many people could die? These are fundamental questions that are epidemiological questions. You can pose them independent of the models, but the models give you a framework for thinking about these questions. And the answers to all of these questions that we realize depends upon one particular number called the basic reproductive ratio. It's called R0. And this, if you remember the equation that I wrote down, they had two parameters, a beta and a gamma. And the dimensionless quantity that you can construct out of these is beta divided by gamma. And that's the essential definition of the reproductive ratio. You can scale everything out in terms of just this quantity in those equations, and then say that diseases differ by their reproductive ratios. So if you looked at numbers here, if you looked at Ebola has a reproductive ratio of about 2, swine flu of 2, HIV of 4, smallpox of 7, and measles is 18. So there are a couple of things that are surprising here. One is measles is such a large reproductive ratio. What's the interpretation of this? The interpretation is very simple. It's given one ill person, one person who's infected in a background of completely susceptible people, how many people on average does that single person infect? So with measles, that you infect an average of 18 people. With Ebola, you infect an average of 2 people. The Ebola number may seem surprising because it's, you would imagine that having heard about it as a dreaded disease, it has a relatively small reproductive ratio. And then the answer is that it's not actually very easy to catch Ebola. Once you catch it, it's very hard. It's impossible to recover from it practically, and you will almost certainly die. The large fraction of people who contract Ebola will go on to die. But the only way to get it is to be in contact with the bodily secretions of an Ebola patient, which is, unless you're a sort of medical worker or a caregiver who has to be very close to that person, typically that sort of bodily contact doesn't happen. The number for COVID-19 sits somewhere between 2 and 3. At least the best that we know now is that it's around 2.2, 2.3 right now. But that number is not only a biological number. It also affects, social factors also enter into it in a way that I will describe a little later. So for the coronavirus, R naught, as I said, is between 2 and 3. But currently it's lower because of measures such as lockdown, distancing, etc. And our current estimate for this number is around 1.55 from the various statistical methods that we've used to estimate this. So the reason that it's such a pliable number is that it expresses many things. The ability of the infection to pass from person to person, which is a biological factor, to cause an infection in someone else, which is biological in character. The ease with which people contact each other, which is social in character. The average number of contacts that a person makes during the period that they're infectious. So you can see that it's fairly complicated in terms of different quantities that actually enter this.
Gautam, one question for you. You define R0 to be a number when everybody is susceptible.
Yes.
So does it make sense to measure it when some population is infected?
No, it doesn't. So then the R naught goes by just simply reproductive ratio. It's not called the basic reproductive ratio anymore. It's called R in that case. So what we do now is estimate R rather than R0. R0 really makes sense only in the beginning of the epidemic, not after that.
What does R mean now when some people are infected?
It means kind of the same thing. How easy is it to transfer the infection from a person to person? But if you estimate it from the number of new cases that you get over the last week or 10 days or 2 weeks, and from that particular time series tells you about what is the rate at which infections are propagating in the population. But it does vary with time. And the sort of secret is once it drops below 1, then your epidemic is over. And the real aim is to try to push it down as far as possible. But it's interesting precisely because it combines both biology and sociology in a sense. And it's really not a purely biological number in that. Okay, one step above that is SEIR, where you can have a susceptible, an exposed, an infected, and a removed compartment. And you put in the exposed compartment in between because you can have a situation where you have someone in which the virus is building up, so they're exposed to the disease, but they're not manifesting symptoms yet. And a big discussion in the community is to what extent is the exposed compartment able to be infectious? Because here you can have a person who is not themselves showing symptoms of the disease. They may not even know it themselves. They're asymptomatic. Nevertheless, they may be able to infect susceptible people. And you don't tend normally to last very long in the exposed compartment. It guesses about 2 days to 3 days. And then you move on to the infected compartment and then to what's called a removed compartment. The removed can either mean dead, it can mean recovered. This is just the SEIR model. And you can already see that we're building in one additional level of complexity in here. And I'll show you a much more complicated model as we go along. So the model that I want to tell you about is a model that came from the Indian scientists' response to COVID-19, a group of people who got together to essentially look at what would be the public impact of COVID-19 to ask if scientists themselves could find a way of communicating to the public issues connected with COVID-19, busting hoaxes for one thing, providing simple, usable messages for the public for another, looking at technical papers and trying to provide a simplified explanation of those papers, as well as do modeling for the disease to see what would be its impacts for India. So one aim was to try and make an India-specific model. So this is the web page of the ISRC. There are two reports on that web page for the modeling part of it, and this represents the work that I will tell you about now. But you can go back and look at it there. All the details are in there. So the aim was to provide a state-of-the-art compartmental model for COVID-19, like the SIR, like the SCIR model, but with appropriate levels of detail, really specifically looking at India and how it's spread in India, as well as the states and districts. So rather than have an India-wide model, which we always thought never made any sense, we thought we would try to make it as local as possible, go from India-wide to statewide to district-wide, and maybe even look at cities and subparts of cities as well in that particular context. The second part of it was to try and look at the rates that entered the model. The SCIR, all of the SCIR equations or any type of general set of model equations that you could write has to have rates in them. These rates are really biological in character. So we wanted to look at biological data and try and fix these so that other people could use them. And this would be a benchmark set of rates that we could use and improve as we learned more and more about the disease in India. We wanted to include demographics and migration as well. But specifically what we wanted to do was to look at interventions that you might make. For example, what are called non-pharmaceutical interventions. A pharmaceutical intervention is like a drug or a vaccine for the disease. It's a pharmaceutical way of dealing with the disease. A non-pharmaceutical intervention is something that tries to alter social behavior and social mixing in such a way as to reduce or alter the spread of the disease. We wanted to make this an online tool and be transparent in how we were updating it, partly because we do see the problem with non-transparency with a lot of models that are out in the literature and people who have been talking about modeling exercises, but not providing enough detail. So now let me describe what the model is so that it can be clear as I go along. The idea is to replace the SEIR by a far more complicated set of models that describes essentially patients' progressions to the disease. So the picture below tells you that if you start with a susceptible and infect them at some point, they all pass through a period which is called the exposed period, just like the SCIR model, but then their trajectories from there can be different. From the exposed period, you can go into a completely asymptomatic period. So that's the first line of the graph below. That lasts for about 5 to 7 days, and then that person gets recovered. For mild symptomatic people, they start off with an exposed compartment, they move to the pre-symptomatic, then they stay mildly symptomatic for a while and then recover. You can be severely symptomatic, which means you start off with exposed, become pre-symptomatic, become severely symptomatic, require hospitalization, and then recover at that point. And then you can have a critical form of the disease, which means you start off with exposed, move to pre-symptomatic, go from there to strongly symptomatic, then hospitalized, and then you bifurcate into either the death compartment or the recovered compartment. So this represents the trajectories, the possible trajectory that a patient might have through the disease, all the way from starting off as susceptible to the point at which they either recover or they die. Here's the sort of compartmental or the box-like formulation of the same picture that I described a little earlier. So susceptible becomes exposed, exposed branches out into asymptomatic or pre-symptomatic. The pre-symptomatic further branches out into mildly symptomatic or severely symptomatic. Mildly symptomatic recovers, severely symptomatic goes to hospitalized and then a death state. And there are various branching ratios that control these and various rates at which you go from one to the other. So what I want to draw your attention to is how exactly susceptible people become infected. For that, they need to be in the presence of infected people. So the infected categories are the IP, the IA, the IM, and the IS. And you can see these dotted lines that are called CS, CP, CA, and CM, which connect these infected categories to the susceptible category. So what we realized was that you could think about intervention in a couple of ways. One is an intervention that affects beta alone, which is a biological intervention. And the other is an intervention that affects the Cs, the CA, the CP, the CS, which could be interventions from outside, which are the non-pharmaceutical interventions. It's some combination of the betas and the Cs that gives you the reproductive ratio, which characterizes the disease. But you can imagine tweaking some of these. For example, if you were able to identify the asymptomatic carriers better than you can currently, you would weaken that connection between the asymptomatic and the susceptible categories. So what we intend to do is to look at the effects of varying these connections, CS, CP, CA, etc., to ask what are the effects on the disease overall. So as I said, the Cs represent the intensity of contacts between any of the infected categories and the susceptible categories. It's a non-pharmaceutical intervention. The product of the C's and the beta determine R. If you reduce R, that's equivalent to a lockdown if you change R completely. If you reduce the C's, that's a combination of testing and quarantining, as we thought of it in this way of imposing a certain intervention on the community. Okay, many generalization. The equations on the bottom right are the set of equations for just the picture on the left, but you can subdivide these compartments. You can make them age structures. So you can have a susceptible population between the ages of 0 to 10, 10 to 20, 20 to 30, 30 to 40, etc., etc., etc. You can, this is important because it's known that more elderly people are susceptible to more violent forms of the disease. So they will be more severely affected by the disease than younger people. And the probability of moving to the death compartment is proportionately higher. The other reason is that you can also put in pre-existing conditions. For example, if you know that 20% of the population between 50 and 60 is likely to be diabetic or pre-diabetic, you can put that into these subcompartments in terms of the propensity to move into one or the other of these other compartments as they move forward in time. The model gets more and more complicated from now on. The first version that we have, the version 1.0, had a model like that for every district level model for the 730 odd districts. So each of those districts is described by one set of these equations, and they're also further subdivided according to age stratification. So it's a very large number of equations that you're solving at that point. So there's age structuring. We also include the ability of people to move from one to the other. So there's migration. There's healthcare accessibility. So you can model the population fraction that cannot access healthcare or hospitals. And what we haven't put in yet is some measure of health metrics that are district specific. So that's the next thing to do at this point. We can also do this for cities. So imagine the top 300 odd cities in India. They're connected through rail and air networks. And you can ask, if I see an infection in Bombay, what is the nature of the spread of that infection across different parts of India? And you assume, so this is one way of putting in migration between different routes and asking, how much later does it happen that something in Bombay has moved to Delhi and Chennai and Calcutta and Hyderabad? And how does the infection spread once it reaches there? Okay, with all of these exercises, there is one universal difficulty, and that has to do with initial conditions. If you wanted to start the simulation off today, you have no idea what are the numbers that you should put in of people who are exposed, people who are pre-symptomatic, or people who are asymptomatic. All you have access to is a number of symptomatic cases that have been tested and declared positive with a COVID-19 test. There is many sort of slip between the cup and the lip on that particular count, because we know that we're not testing enough. We know that people are not naturally disposed to go and report to hospitals if they think they're going to be quarantined or being, they don't want to deal with the complexities of the hospital system in India. So you may not be seeing patients who generally require medical attention, especially if their symptoms are mild. You may not see the asymptomatics at all unless you test them and they happen to be the vicinity of close contacts of someone who's already symptomatic. So there you have to make guesses for factors of 10, factors of 5 between these real numbers, and try to do as best as you can in terms of fitting to data. The reproductive ratio, as you asked, we take it from statistical models that try to build it up from the number of cases across a certain time interval of, say, 2 weeks currently at this point. And this is encoded in models called IITB. There are also Bayesian models. So this is what the Imperial College uses in its suite of programs that they put into their own codes that do the compartmental model types of things that we do. So we've checked and validated all of these things. So if you want numbers, the number as of 6th May for India, our number is 1.55 for the reproductive ratio. For 24th April for Maharashtra, it's 1.54. And I will use that number a little later when I do a Maharashtra specific calculation. Yeah. So what I'm going to do, I'm going to choose some examples. I'm going to choose Maharashtra as an example. I'll also do Tamil Nadu, Chennai, Pune to illustrate different things about what we can do. So we start with Maharashtra, 11 crore population, 100 exposed, 10 infected persons, and we divide them in some way between 7 asymptomatic and 3 symptomatic, just to make a point. It's not as though these are the numbers that are really current numbers in any sense at all. We assume there's no initial hospitalized, dead, or recovered. So we start counting our hospitalized and recovered from that particular point. We model a lockdown by saying that your R plummets from a value of 2.4 pre-lockdown to a value close to 1. And less than 1, the epidemic dies down. So this is equivalent to saying that the lockdown has managed to contain the epidemic because of your ability to pull out patients, isolate them before they can spread the disease to other people. We impose a lockdown on day 30, a single lockdown lasts 30 days. We've tried various things with periodic lockdowns, open breaks, so a lockdown for 7 days, a break for 5 days, lockdown for 7 days, etc. The important thing about this, and this is really going to be the main theme of this talk, is the way we impose what we call quarantine testing, which is the ability to test people, even if they happen to be asymptomatic. But this is like envisaged as a test of the general population, which is slowly ramped up. So in the beginning, you might only be able to test 5 or 10% of the population. But you imagine that 6 months down the line, that number has increased to 50%. 9 months down the line could be 80%. So we assume a sort of exponential increase in your ability to do testing. And you apply that in this particular case. The only slight twist is, because it's much harder to find asymptomatic people than it is to find symptomatic people, the time scales over which you can identify asymptomatic people is assumed to be double the time scale which you do for symptomatic cases. So that was just a modeling choice, and we could change that later if we wanted to. Okay, so first of all, do we reproduce standard answers? And the answer is that the initial number of cases that you start out with just doesn't matter, and this is known. All that initial number of cases does is to shift the location of the peak of the epidemic, but the peak value remains the same. So whether you start from 10 to 100 infected individuals, you can move the peak of the epidemic from about 80 days to 90 days to 100 days, but that's all that it does. So this would be a canonical number for no interventions at all, where you would have around 200 lakhs in Maharashtra infected at the peak of the epidemic if you did nothing at all. No non-pharmaceutical interventions, no interventions of any sort in pushing the number of R0 down. If you change the R0, what it does is to flatten the peak. So this is, again, very canonical epidemiological knowledge. By changing R0, which is equivalent to changing beta in our program, you reduce the peak of infected people. You spread that peak out over a larger time. And this is really what any non-pharmaceutical intervention tries to do. Its effectiveness is measured by the amount to which you can push down that peak, thereby reducing the stress on the healthcare requirements that you might be expected to provide. If you now do testing and quarantining in the way that I described it to you, and you do this, imagine a testing regime that is fast, that ramps up to essentially perfect testing in 100 days or 150 days or 200 days or 300 days, you can push that down. So the background that you can see in the slide is the sort of grayish part of it is the no intervention scenario. You can see just by intervening with quarantining testing, as I described it to you, you manage to push that number down. So it goes from about 200 lakhs to about 80 to 40 to 20, depending upon the nature of that intervention and how fast you can ramp it up. And you see a similar decrease for the hospitalized compartment as well. So increased testing and quarantining reduces the peak. If you put a lockdown in, all that does is it shifts the peak. It doesn't do anything to the absolute number. So you can do a lockdown with quarantining and testing. You reach a certain peak. Whether you have the lockdown or you don't have the lockdown, all that it does is to move the peak around. If you have a single lockdown, but then you continue quarantining testing all the way through, what that does is again give you a substantial reduction. So the lockdown helps you identify cases, push the number down, reduce the transmission within the community, and your quarantining and testing enables a further push down and a further shift to the right of that. So now you're talking numbers in the region of about 5 to 20 lakhs compared to 200 lakhs that you had earlier in terms of the total number of infected. You can also do more clever things, such as what is called a light switch lockdown. That is, you impose a lockdown if you cross a threshold number of infected, let's say 500 infected in this calculation. Along with that, you do your quarantining and testing. And once you fall below a certain number of infected, you can relax that and wait till you cross back again. You can do this. It turns out to be not extremely efficient compared to other methods of doing it, but you can still reduce the peak to still a manageable number. If you do a periodic lockdown, which is open, close, open for 7 days, close for 5 days, etc., you can see these little wiggles that are the numbers of infected going up, together with quarantining testing, that again is a substantial reduction in the number of infected. So now you're talking about 0.2 lakhs to 0.3 lakhs numbers of infected. Here's one other example of what you can do in one city, for example, Chennai, with quarantining and testing. So the no intervention amount of the hospitalized population is 175,000. With quarantining testing over even a year and small variations in the numbers of people initially infected, you can push that 175,500 right down to a number like 50,000, and even further down if your number of infected isn't too large to begin with. So we look at all the way between 100 and 500 infected in the beginning with appropriate multiplying factors for the people who you cannot see, the asymptomatic people. You can, the more sharp and the more targeted you make your quarantining testing, the better it is. if you manage to test a substantial fraction of your population within 120 days, which is 4 months, you can push the number of hospitalized down to a number like 400 or 500. So all of this, what you can do with these numbers really relies on how efficient the quarantine and testing regime is. That's something that comes out very naturally of these equations. We've been asked in more recent weeks about what is it that you can recommend regarding staggered lockdown measures applied to a particular city. We said, take a city and imagine that you can have some people going to work, some people staying at home, or everybody going to work and everybody going to home, along with some fixed periodic schedule. So they go to work, let's say 2 days a week, so work, and then for some others, 4 days a week, they stay at home. So it's work, lockdown, work, lockdown. So you can work for N days and be locked down for N days. So that's one way, which is a periodic synchronous lockdown. The other way to do it is to divide the population into, let's say, 3 parts. Have the first third go work for the first 2 days, then come back and be locked down. Have the second third go for the next 2 days, then come back and be locked down. And the third goes for the next 2 days, and then come back and be locked down for 4 days. So now everyone goes back to work, but the amount of people at work is staggered. You have 1/3 of the number of people at work at any given time. And you impose the lockdown on those who have come back who are not at work fairly stringently, thereby keeping R naught down. This is an idea that was originally motivated by Yuri Alon and group, who also have been looking at situations like this. And if we find that if you do that together with testing and quarantining, which is something that Alon's group didn't do, you find that this is an extremely efficient strategy. So the number with asynchronous lockdown, where people go back to work, and then the numbers of infected, of hospitalized, can be a number as small as about 400 to 500. If you combine an on-off asynchronous lockdown for 1/3 of the population, together with testing and quarantining strategy, even if that is spread over a fairly longish period. So we really believe that this is a good way for people to return to work. Of course, this statement comes with many caveats. So it comes with the caveat that you have to ensure that people under lockdown situations are really under lockdown for the 4 days that they remain at home. And that people at work maintain social distancing so that they are there, but then even since they're there at relatively lower density, 1/3 density than they would normally, it's a little easier to do that. It's a little easier to monitor and check that. We can put in age structuring, and the figure below shows you the changes in the hospitalized number with and without age structuring. And you can see that there's a slight difference in the two curves. So the more older your population is, the more significant the increase in the numbers of people who have to wind up in hospital because they have more serious forms of the disease. Here is some real data for Maharashtra, where we sort of imagine, we took that number on the 24th of April, our own calculated number, and we extrapolated. So this is a bunch of curves for different initial conditions, for exposed to 5,000, exposed to 10,000, exposed to 2,500, which is the lowest curve there. An initial amount of the asymptomatic of 450 and a pre-symptomatic of 440. And you can see that that little dot, that circle that you see there is the Maharashtra number today. And that would be the extrapolation that you might expect, around 10,000 or 11,000 as of 2 days ago, I think that was. And you can see where it would go if you extended that curve beyond and you lifted the lockdown and you went back to a different lockdown, a working figure for R0 of 2 and a lockdown figure of 1.5, which is what that increasing curve actually signifies. We've also done this exercise in a much more local way. We did this for Pune, for different areas in Pune, different localities, Hadapsar, Bhawanipet, Kondwa, etc., where we looked at the effects of having a lockdown moving to a staggered lockdown. So you can see the peak coming down and further down. So estimates of hospitalized fractions in these different areas based on this approximation that we can make, that a staggered lockdown is probably the best way to get people back to work while ensuring that the disease doesn't spread too far. So right to the end, let me talk a little bit about some work we've been doing on more realistic models for disease spread. So those compartmental models, you should think of them as essentially first pass models to gain intuition for what might be happening. But we've been doing similar agent-based models. So this is for a population in a sort of dense area in South India with a bunch of collaborators. And that's what it looks like. If you sort of looked at a map of this, it's a bunch of villages placed together. And this is actually a movie which I can try to run for you. And you can see people moving back and forth. So this is a movie in which you have people infected, transmitting the disease, but they're still allowed to move back and forth. So there's little dots that move back are the actual people themselves. This had about 3,000 agents. We are now doing 120,000 agents because that is the population that we have to work with. So they come, they get infected, they can move to hospital, you can monitor hospitalization numbers, etc., as time goes on. So an agent-based simulation that we're doing now is for this particular community. We have 130,000 agents. Initially, what we wanted to do was to study lockdowns in dense urban situations. So we took 2 exposed people right at the beginning and began the process of infection. We began running the agent-based code here. They were all confined to homes in the strictest version of the lockdown. So the only person they could infect were the people they were at home. Since we have many details about the study population, we know how many people stay at home, where the homes are located, et cetera, et cetera, et cetera. So we know how many people are being infected. So the rule is that the moment someone turns symptomatic, they're pulled out, the whole family is tested, and the whole family is quarantined if anyone else is found positive. If someone is seriously ill, they're taken to hospital, and hospital numbers are also monitored, and everybody in the family is picked up and tested. We also varied the adherence to reporting. So we said that assume that 20% of people with mild symptoms will report, whereas 80% of them will not report. So that's a not very adherent to reporting scenario. We could also look at the situation where 80% of people with mild symptoms reported and the rest didn't report, and as well as 100% of them reporting, and as well as none of them reporting, etc. So then we tried looking at various relaxations. For example, we said, as all of you know, I think, it's very hard to keep kids confined in a small location for weeks on end. So imagine what would happen if the children went out and played with other children on the streets in front of the houses, not allowing them to go far, but just stay on the streets right in front. What happens if you have places of worship and people go to places of worship but nowhere else? They stay at home or they go to places of worship thrice a week for, let's say, about a few hours each. What happens if children go back to school while the parents remain under lockdown? So children go to school, parents come back. That would be admittedly a somewhat unusual way of doing things, but that's another possibility. So let me just quickly summarize what happened. In the situation where you have 2 people to start the infection off, and everybody only stays at home, the maximum number rises to about 4 to 5 people of people infected. If you allow the children to play, along the street, that number rises to about 80 people. If you allow for religious places and people to go every other day to a religious place for a few hours, that number rises to about 2,000 people. Remember, the whole group is about 130,000 people. The moment you allow children to go to school, that number goes up to about 33,000 people. And you can see a whole bunch of gray, that these are all different trajectories. And the solid line that you see there is the average trajectory out of all of this. So again, the numbers are 2 initial exposed, maybe about 4 to 5 infected, actually not even 12. Children allowed to play in the streets, about 80 infected. Place of worship, 2,000 infected. Children going to school, 35,000 infected. The reason for thinking about this is that the issues that Bombay faces, for example, are how do you deal with people in extremely crowded scenarios? What is the nature of social behavior? How do you relax the constraints that are imposed by the fact that people have to live cheek by jowl in highly dense situations over a long lockdown period of about 30 to 40 days? What are the effects of weakening that lockdown in any way? These are the questions that we wanted to look at. This is all a very ongoing work. This is very preliminary results. So let me sort of wind up by telling you what we've been doing, what the main points that I want you to take back are. The first is the importance of testing and quarantining, which I think comes out very cleanly in this calculation. It doesn't matter even if you ramp up testing slowly over 6 months to a year, there is still a profound importance to actually having that testing in place and being able to do it to identify cases. The staggered asynchronous lockdown seems to be a way to open up post-lockdown. And this again is something that's worrying many people. What are the economic consequences of having people not go to work for long periods of time? And we suggest that the staggered asynchronous lockdown, at least applied in regions where the number of cases is not increasing very sharply or has remained stagnant or is going down, might be a solution to that particular problem. The INDSCI-SIM model is a detailed compartmental epidemiological model for India. It's probably the most detailed model that is out there. And these models are very good, as I said, models to ask questions to about strategies and methods for intervention. And because they can be run fast, because there's a lot of information that you can put into them that is epidemiological in nature, you can hope to get answers that are sensible and be able to compare different strategies and scenarios. You need age-based models to answer much more local questions. What, for example, does the train network do in Bombay, as opposed to another city which doesn't have such good public transport? You can ask for specific place-dependent insight into how the disease might spread. As always, you need better inputs, you need better data, better statistical estimates for all of these quantities. So let me just finish up with sort of, just for my sort of thank you slide. The fact that the INDSCI-SIM model or the epidemiological compartmental model is a deterministic model. The agent-based models are stochastic models, and they're also geographic models. They put people out over a geographical space, and geography is important. There's a slew of models in between, which we've been working on, which again, I think is an area that we really want to spend more time on, which is to look at Gillespie-based models where you can put in details of networks, individual variation, the progression of the disease, etc. These are one more step more complicated than the deterministic model. They will help you answer some questions. The other thing that we really need is better statistical models and data analysis, especially data analysis. Data that is coming out of government is pretty much untrustworthy. And you have to be able to piece it together from newspaper reports, various types of other sources, along with whatever government provides you. The states are doing a better job than the ICMR currently of providing you good data. What you need is statisticians to look at the data, to put in all of the right time delays, all of the right Bayesian priors for reporting of data, etc. You need health economists, you need sociologists to tell you what is the nature of contact. So what goes into an agent-based model is really sociological information. Who interacts with who and how strongly? How many contacts does a person on a specific job have with other people? So with that, I hope I've taken about 35 minutes, I think. So let me thank a whole bunch of collaborators. All of those in yellow on the top are people who were part of the INSIGHT-SIM initiative, and Dheeraj and Snehal and Balchandra did a lot of the heavy lifting with the code. Vinod Kumar, Abel, Farina, and the IMSc group is working on the agent-based model of the population that I told you about, the slum population. The ThoughtWorks team that's a very large agent-based model which we want to try for India. And the intent is not just to look at COVID-19, but to look at larger public health issues across the country and to see what information we can put into that model that will be useful for planning for both economics, public health, sociology, a whole range of interesting things that we've been wanting to do for a while. So that we've already made in the meeting on Saturday, the ThoughtWorks team made a presentation for Bombay and what they were doing with what we've been doing with Bombay. And that's an additional, that's part of this much larger scope of Bharat SIM, as we call it. It will be an agent-based model for 1.38 billion people, and to see what information that gives us. And of course, there are many others who I should thank, including many people in this audience, people listening to me, who've contributed to my understanding of these issues across multiple years. So thank you so much for listening, and I'm happy to take questions.
Thank you. So anyone who has questions, please feel free to jump in.
So, Gautam, in the asynchronous lockdown, so the assumption is that people who are supposed to be, who are not supposed to be in the working group currently, they stay at home, and
they stay at home. They stay at home under lockdown conditions.
Okay, so and this is assumed for all people, right? I mean, there's no, I mean, or is there a compliance that can be taken into account or something? Like if there are some people who tend to violate this.
We could do that. We haven't done that. We haven't tested it. That's a very good point. What happens if you have people who don't, if you have some subpopulation that doesn't obey this, and what would that do? We have not looked at that question yet, because we usually tend to look at ideal situations and then sort of say that the measures should be such as to ensure the ideal situation and not allow. But you're correct that in that these sort of nice estimates will not work. We don't know yet as to how bad it will be for the overall scenario.
Okay.
It's a good question. That's certainly something we should look at. Thanks.
These periods 2 and 4 and 5 and 7, have they been optimized?
We looked at them. We looked at a whole bunch of 2, 4, 4, 6, 5, 7, etc. There didn't seem to be a very large difference. And basically, small changes in your testing, quarantining procedures can account for that. So we didn't make too much of a point. I mean, it sort of makes sense that they should be related to the underlying biological time scale at which you move between these compartments. But remember that this is a compartmental model of fair complexity. There are many different time scales at play. So exactly how to disentangle that, we haven't quite figured out. And the model that Uri Along works with is a much simpler SCIR model, where the number of time scales is smaller. So presumably these time scale effects should be there, but at the rate of 2, 4, 4, 6, 4, 8, etc., we couldn't see that.
Hi, Gautam. Vijay here. In the agent-based model that you showed, when you let the children go to school, the numbers really shot up. You had an age-based difference in the infection rates with children? Is that
no, we didn't. In fact, it's currently understood that children, okay, so this is very controversial. But do children infect other children at the same rate that adults infect other adults? There are two sort of two views which take completely opposite points of view, that the viral load in children is the same as the viral load in adults. And the other says that children aren't particularly efficient compared to adults in transferring the infection, which I really don't understand. If the viral loads are similar, given the extent of children contact in the school, it's sort of hard to imagine that they aren't very effective vectors of the disease, which we know certainly to be the case for influenza, for example, which is again very like this. And the reason for this seems to be that the more you allow it to spread, as long as you allow children to move only along the street, they're only infecting people along that street, but they're not infecting the next street. The moment you allow people to go to a place of worship that is common to that particular village, you're ensuring that all of that village, anybody who goes there and then goes back from there, can be infected. You take children to a school, and we know every school that these kids go to, they come from all of these other villages around there. They come to a particular school. So you've suddenly expanded the scope of the disease. So really the message from that, I mean, it's not a very deep message. It's just that the more local you can keep it and prevent different subunits from interacting with each other, the more you can hope to control the disease. But it's nice to see these things very specifically illustrated in the context of a very detailed model, which we have a lot of information for.
Hi, Gautam. Prasad here.
Hi.
Just a quick question for you. Because now in your model, you have a coupled ODEs.
Yeah.
So, because there is migration from one state, one state to other state, or even between districts.
Yeah.
It's always, why don't you see secondary infections again starting up? I mean, why it's always all the curves are first going up and then infection?
Because the curve that I showed you didn't have migration, but the picture for India that I showed you with sort of colors all over across the cities, that had migration. And there you can see very clearly the secondary effect. You can see Bombay starting off as a big red circle, and then suddenly Delhi takes over, Chennai takes over, and you can see it spread over all the 300 odd cities. This is actually available on the online site, so you can actually test it out to see how this works. But the data that I showed you was for one geographical area with no mixing from others.
Hi Gautam, so Abhishek here.
Hi Abhishek.
Yeah, so I had a question on the way you introduce this testing, quarantining as an intervention.
Yeah.
So, so basically, if I look at the reproductive number, I mean, it's essentially given by beta. Let's say just for SIR.
Yeah.
Then it's given by beta times C divided by lambda.
Yeah.
Right. So now, so by social distancing, it seems what you can do is you can change C essentially.
Yes.
But when you do testing quarantining, it seems like what you do is essentially you remove people from the infected population, right? And so that should, it seems to me that you should be really changing lambda.
So that's another interpretation that you could have additional compartments and you sort of test them and you move them out so that they don't infect another. The interpretation that we're saying is that anything that weakens the connection between two compartments can also be thought of as an intervention. Any testing quarantining which removes people from one compartment overall weakens the connection with these two compartments. It's a question of interpretation, I think. But that's a good point. I mean, in principle, the only problem is it introduces more compartments. If you wanted people to move out from there into quarantine compartments, thereby reducing the numbers in the infected compartment, that would be also implementable. It's just more complicated. But that's a valid point.
So why do you need compartments? Because I mean, because you just care about the fact that the number of infectious people are going down, right? But where I mean, you don't care about where they're going. So I mean,
no, that's correct. So it enters as a C times the I times the S. So C times the I can be interpreted as that fraction of I which interacts with S. So a C that is weakened with time is like an increasingly small number of the I people in that compartment that is able to infect. So the interpretation is again precisely the same. I could choose to do it in another way, but this interpretation just said there's a fraction of the I's which can infect the susceptible, and that we interpret the quarantining testing as reducing the fraction effectively.
Okay. But I think, see, I mean, like, I mean, when I think about it, it seems like if you really want to connect with, for example, things like the number of tests done per person and per whatever rates, then it's more natural if you like include it in the lambda.
What is the lambda that you're talking about? That the exit rate from that.
The rate at which number of infections, like I to, let's say, R or the other compartments.
Because
So do you have this
Yeah, you could do, you would have to introduce another compartment because you really want to keep track of people, how many people are in hospital or how many people are asymptomatic. These are things that you want to keep track of. So the only way you would do it would be to allow that to happen, but to pull out people into another compartment, which you call the quarantine compartment, leading to an effective decrease in the compartment that you want to study. So you have quarantine people, the rest of them go on to the hospitalized, etc. stage, especially for the more severe people. From those stages, they don't infect the susceptibles. It's only in the in the sort of infectious stages that they can actually infect. I think it's the same thing. All you're saying is that there is some fraction of the infected which can infect the susceptible, and that fraction is controlled by quarantining testing. I mean, it's a question of which you're more familiar with, but I think it's an equivalent interpretation. But we can maybe discuss this.
Yeah, we can discuss later.
Sure.
Maybe.
Yeah.
For India level, how did you calibrate for migrations?
So migrations is, what you do is that you assume balanced migration. That is, you have a whole bunch of categories there. They can, between I and J, you ensure that the total population in I and the total population J don't change. So you can set up an iterative scheme that ensures that when you solve these equations, that's not the same as having, people being really moved from here to there, which decreases the population here, increases them there. We're working on that with what are called gravity models for migration. And we would like to get better social data for migration, which we could put in, but have not quite accomplished that yet. There's very limited data that we know. In fact, IIT Bombay, the people who gave a talk on Saturday, mentioned some migration data for districts in Maharashtra that they had. And there probably is some old social data from the 2011 census or after, but we don't know how comprehensive it is. And that's something that we really would like to know, be able to put in. Because now it's not very clear now that people are moving back, whether they will come back again, what are the flows we expect, what is the time scale of those flows at all, we really don't know.
On migration, the census data contains actually very detailed data on migration. It's called table D3.
Okay.
It has so it doesn't have one particular thing. So I was looking at it for other reasons, and there's one particular thing that it doesn't have, which they have the data, but they don't release it publicly.
Okay.
What they do release publicly is something like this, that for every district in every state, I can figure out how many people from any other state are living there, for how long, and for what reasons.
Okay. But this is still 2011 data, which I guess should be...
This is 2011. Yeah. And 2001 also. So both census data have...
Okay, yeah. No, that's certainly something that's important, and we would really like to sort of, use that information sensibly.
What don't release publicly is the other way around, which is to say, for each district, if I wanted to know how many people from Lucknow are living in Mumbai, that is...
Okay, I can imagine that's sensitive information.
How many people, how many people from UP are living in Mumbai.
Okay.
Gautam, what's your contact with the policymakers? Are you able to influence them in some way with this research?
Certainly not directly, and I don't think there's nothing that either they or I would aim to. But I think the fact that some of this work is being reported, there are informal channels, and I sort of don't know what happens to it after that. We've been asked informally sort of semi-formally by Tamil Nadu government, the Chennai people, to tell us. By Pune, there's some bit of informal connection that we have a group of scientists who work with the Pune Municipal Corporation. At the larger scale, it's impossible to figure out what is actually going on. So one just hopes that, certainly people like Vijay are very much on the ball, and Vijay knows exactly what's going on. So hopefully some of this information is percolating up. Although with this government, one is not very sure who they're listening to or what they really understand. Sorry to be pessimistic, but this is about what I understand.
So if there are no more questions, we'll thank Gautam for a nice talk, and we'll move on to the next speaker now. Our next speaker is Manoj Gopalakrishnan. So Manoj, do you want to go ahead and share your screen?
Yeah, I think Gautam will have to...
Yeah. Sorry. Yeah, I just should try to figure out how to do that. Yeah. Apologies. Yeah. That work?
Yeah, that's fine. So go ahead, Manoj. Yeah. Well, what's nice about today's speakers is that both Gautam had a connection with TIFR. He spent 2 years here as a visiting fellow, and Manoj was a faculty here for many years before he moved on to IIT Bombay.
No, more than that, I'm a adjunct professor at TIFR at the moment.
Is that right? I see.
In DBS.
So, connection, wonderful. So Manoj was, I guess, in TIFR for many years. Now he's moved to IIT Bombay. His interests have always been in kind of, using mathematics and biology in variety of forms. And now he's doing very interesting work in pooling samples. And I guess we learn about that from him today. Manoj?
Thank you, Sandeep. Yeah, hello, everyone. And this is my first time giving a seminar on the internet. So it's a little weird. I just had a power outage and I moved to my phone 3G network. I hope it stays up through the talk. We will find out. Yeah, so thank you, Gautam. That was a really interesting sort of talk. I think it is one layer sort of higher than what we are doing. If the sense you got, like I did from Gautam's talk, was that it's very important to test more people, then I'm going to tell you how that can be accomplished, and at least one of the bottlenecks in testing, how it can be eased a little bit, and we can test many more people and strategically do something about increasing testing. So here is the goal. The goal is we want to ease lockdown. So we cannot ease lockdown unless we start testing people who don't have symptoms. Okay. Right now, we are finding it hard to test people who have symptoms, forget testing people who don't have symptoms. So how is that going to be possible? So first of all, why do I say this? Everybody knows this. COVID-19 carriers are asymptomatic for many days. So they are carrying the virus, they are spreading it, but you can't see it. And those who are not identified can be super spreaders. In India, at least, test kits are running out. And of course, recently there have been many more companies coming in and increase in the amount of test kits available. But if you do want to increase your testing exponentially, like Gautam was saying, then yeah, you need to, I mean, in practice, you're still running out. So I know many testing centers are still running out of test kits. Yeah. With limited testing, there is no alternative to complete lockdown. There is no alternative to economic hardships. Okay, maybe there are alternatives. You can live with the virus and take whatever consequences that has. So what is the solution? The solution is to try to get more from less. And there are multiple ways of doing this. The most common way of doing this is, you may have heard of this maybe in the press or you have thought about it yourself, is pooling samples. So the idea is that most of your tests are wasted. You're doing 100 tests and you're getting maybe 98 negatives and 2 positives. So 98 of your tests are wasted. And if you had somehow magically known that you could pool those 98 samples together and then done individual tests for the other 2, you only need 3 tests to get 98 negatives and 2 positives. So in principle, there is a lot of savings to be had. The question is, how do you get at those savings? And what are the sort of trade-offs involved there? Yeah. So our goal with the technique that I'm going to talk about today, called Tapestry pooling, was this one. We started with this goal. We want to catch 10 positives in 1,000 samples and do only 100 tests. Yeah. So 10, 100, 1,000, that was our goal. And I'm glad to say that, the numbers are sort of, I will show you some results and you will see that we have actually reached this goal. Yeah. So unlike traditional pooling, Tapestry is a single round testing, and there will be savings not only in kits and effort, but also in time. Okay. So what is pooling? So this is sort of the simplest idea of pooling. You put a bunch of samples into one tube. Now, if any of these individuals are carrying the virus and the sample then contains the virus, then the test that is performed here, the most common test, is something called PCR, polymerase chain reaction, which exponentially amplifies the part of the sequence that is unique to the virus that we are trying to detect. So it runs through cycles. So there are you put it in something called a thermocycling machine, which goes up in temperature, goes down, goes up, goes down. So each of these temperature going up and down is called a cycle. In every cycle, you roughly expect that the number of these molecules doubles. And as we all know, exponential functions grow very fast. And after a point, the number of those molecules becomes sufficiently large that you're able to detect it with some kind of a reporter molecule, like a fluorescing molecule or something like that. Yeah. The problem with this naive idea is that you put n people into one tube. If any one of them is positive, you get a positive result. If all of them are negative, you get a negative result. That's the good case. You can send everybody home. But if one of them is positive, you don't know who is positive. Okay. So what do? You do this. You do a 2-round pooling where you take all the tests that came negative. Everybody who was in that test can go home. Now the test that came out positive, you test every person in that in a separate test. Yeah. So this is the kind of pooling that ICMR has already rolled out in many places. Kerala, I think, in India was one of the first to implement such a scheme. And yeah, so this has savings. This has sort of immediate savings, and this is being deployed, should be more widely deployed than it is. And what this can do for you is that for 1,000 people, you can test 1% infected with 200 tests. Okay, this takes 2 rounds, and 2 rounds is not so great, because 1 round of PCR testing tends to take about 4 hours. So then if it's 2 rounds of tests, then you have to do about 8 hours of work. You have to go back to the original sample, which is highly contagious, wear all your protective equipment, handle that original sample again, purify the RNA for each of these, and then sort of do the process again. So there is some effort required to do this, and it has been, it has not caught on as much as one would like. So I think there has been some reluctance to do in the field to do 2-round pooling, because they don't want to sort of handle the same sample again and so on. There are concerns about the effort required to do that. This is especially true in a country like India, where the RNA extraction process is also not mechanized. So there is a process of taking the sample and then washing away the fat and the protein and keeping just the RNA. And even that process often has to be done laboriously, in Indian labs. And that has been also a bottleneck for doing these kind of 2-round protocols. Tapestry is a single-round protocol. What that means is that each sample goes to 3 tubes. Each sample gets tested twice, and each tube now gets samples from multiple individuals according to some matrix that we come up with, based on which test is positive and which test is negative. We can tell who is positive, who is negative. This idea is called group testing. It's been around for a long time. We can do a little better than that in Tapestry, as I'll show you. So here is the idea sort of more graphically. These are a bunch of individuals. Some of these are positive, some are negative. Most of them, let's say, are negative. This is the matrix that we designed, which says which sample goes in which test. The tick marks describe that. So in our schemes, all the columns tend to have 3 ticks in them. Yeah. So each sample goes to 3 different tests. Now, a group testing idea is to look at who is positive, who is negative. And if a particular sample is positive, you would think that every test that the sample came up in would come out to be positive. If a sample is negative, then one would hope that at least one test that the sample occurred in turns out to be negative, in which case that sample can be eliminated, right? So for example, this sample, there is a negative here. So this sample is definitely negative. Yeah. Now, because this sample is eliminated and maybe all the other samples are eliminated here, that might mean that in a positive test, there is only one surviving sample, and that sample can be declared to be positive. Yeah. So these are very simple Boolean algorithms. But in fact, PCR is not just a Boolean algorithm. It doesn't just tell you whether a test is positive or negative. It is a quantitative algorithm. So not only do we have something like this, we actually have a bunch of linear equations where each sample has a certain number, certain copy number of the virus, and this matrix multiplies with the vector of copy numbers to give the vector of test results. And this matrix, of course, is short and wide. So you just by linear algebra, there are many solutions. There isn't a unique solution. But it turns out that if we design our matrix carefully, then the number of sparse solutions you can promise that the number of sparse solutions will be very small, and in fact even unique, if the number of tests is at least k log n, where k is the number of positives and n is the number of samples. So you see that going from n tests, you have gone to a logarithm in n times the number of positives. So there is potentially a big savings here to be made. if one uses a method like this. Yeah. Okay, so to say this in words, each sample is tested multiple times. The quantitative viral load in each pool is reconstructed using the qPCR test. This is what happens in every test. Usually this is ignored and you just return whether it's positive or not. This is the vector of y1 to ym. The viral load of each sample is again some unknown vector x1 to xn. We have some matrix A that we have designed, and A times x equals y is the equation we want to solve. Now, we don't solve this equation exactly. We solve it with some noise model because, of course, this is a noisy process. And in this case, it's not an additive Gaussian model. It turns out to be some kind of multiplicative noise model. So, in fact, we have come up with a noise model for PCR. And using that noise model, we try to, so we have to solve three problems here. We have to come up with a noise model. We have to come up with a way to design this matrix A. And we have to come up with an algorithm for reconstruction. Yeah. And so there is an entire field in sort of signal processing known as compressed sensing, which looks at problems like this. There are many unique features to the particular problem we are trying to solve, which have to do
17,000 year to find the.
Can you just mute?
Yeah, sorry. I had to close a door. So yeah, there are so there are many features of this problem in our setting which are new, and so we can't just use algorithms out of the box, but we have to do some algorithmic work here as well. Yeah? Okay. So how does it compare with simple pooling? So this is pretty much the same. So both simple pooling in 2 rounds and Tapestry pooling will tell you which individuals are positive and negative. Here in simple pooling, you have the same false positive and false negative rates, more or less. In our case, the false negative rate is more or less the same, maybe slightly better, because in fact, you're testing each sample 3 times. So if there was some error in one of the tests, you can still, by doing the right algorithms, you can pick out that there was an error, and that's the more likely explanation than that the sample is negative. There is a conservation of extraction and testing kits in both cases. In our case, the numbers work out better. We seem to be, sort of requiring half the number of tests, at least in the simulations we have been running and in some of our real experiments. Big advantage with simple pooling, it's already deployed in USA, Germany, Israel, now in India. So it's in the field, people are already comfortable with it. This is a new idea. Tapestry pooling is still a new idea. We are, I'll show you some experiments we've already run, and we are in clinical trials right now. The amount of pipetting you have to do is slightly, so each sample gets pipetted once in the first round, and the positive samples get pipetted once in the second round. So that's n plus k is the amount of pipetting. Here the amount of pipetting is 3 times n, and the time to do that roughly scales about 2 to 3 times. Yeah. This is a multiple round thing, as I said. So it requires a second round of RNA extraction as well, which slows things down. Time to result is longer, and the effort required also can, especially if there is no automatic RNA extraction, the effort required can be more. Another big advantage with Tapestry pooling is that it is viable at higher prevalence rates. So 10% prevalence and so on, we can still do Tapestry pooling. Simple pooling sort of doesn't give so much savings at higher prevalence rates. Yeah, it reconstructs viral load. So if there is any clinical sort of meaning to viral loads, then that can be used. We don't know yet, because most of the time people don't collect viral loads. There are 1 or 2 papers and news articles correlating viral loads to a couple of things that have some clinical relevance. But again, as Gautam said in his talk, it's not completely clear. It's not that symptomatic people have higher viral loads or something like that. Yeah? Okay. Yeah. So yeah, the biggest challenge here is it's a more complicated scheme. It's a newer scheme, and pipetting time is a little more. So these things have to be taken into account. Yeah. Okay. So here are some numbers. So you see that we can do 70 samples in 21 tests. The pool size looks like about 10 samples go into every pool. Our pool sizes are going to be slightly bigger than for simple pooling. Yeah, the amount of pipetting is always going to be 3 times the number of samples. Yeah, and this can tolerate about 5 to 6% prevalence rate, and so on, right? So you see, we can do like some almost 1,000 samples in 90 tests and catch around 10 positives or so, with a pool size about 31 or so. Okay? And yeah, the number of pipetting is always 3 times. Okay, we did some experiments. So this particular experiment was done at INSTEM NCBS in collaboration with a lab there. So here we tested 40 samples, and either 0 or 1 or 2 of them were spike. We did not know how many would be spike positives, and we were given the Y vector for each test, and from that Y vector we had to reconstruct how many were spiked and which were the samples that were spiked. So the samples were tested in 16 pools, okay, and these 16 tests were performed. So we were given a vector of length 16. Yeah, we took this vector and we managed to reconstruct correctly all the positives. So all 120 samples in this experiment were correctly identified. Okay. Yeah. So another experiment, this was done with our collaborators at the Wyss Institute at Harvard. So we took another matrix, which was 24 cross 64, and the 64 samples here in this case were, so this was actually DNA of the same sequence as COVID-19. And that DNA was put in 24 pools according to the matrix, and tested using PCR. We got back the test results, and again we were able to correctly reconstruct the positives and the negatives. Yeah. Okay. So these are more recent experiments where this is again at the Wyss Institute at Harvard. We put 120 samples in 40 tests with 3 positives, and the algorithm could identify all 3. And most recently, we put 1,140 samples. So these samples are the entire COVID-19 RNA genome, but synthetic. You can buy this. And these were spiked into mock samples and for the positives. So 11 tubes, they had these positives, and these were pooled into 90 tests according to our scheme. And the pooling was done using a liquid handling robot. The whole process took only 18 minutes. And our algorithm could correctly identify all 11 positives. And we in fact returned a list of 14. So we got 3 wrong. We returned 3 false positives. Okay. So what are the guarantees here? So besides estimating the viral load of each sample, we also do a bunch of other things. So the first thing we do is we estimate sparsity. So of course our test is designed for a certain level of prevalence rate. But what if the batch that we got has a much higher number of positives than the prevalence rate? We should first be able to detect that such a case has happened, and we do this. We have an estimator for the sparsity, which is actually very tight. So we will immediately know if the number of positives is very large. And in that case, as soon as we know that number of positives is large, we can in fact, we will always return these 3 categories, right? Positives are confirmed positives. You don't need to do a second round of testing. Negatives are confirmed negatives. You don't need to do a second round of testing. And undetermined are the ones that will usually be an empty set. We will not have undetermineds if our assumption about the prevalence rate matches the number of positives. If we get an unexpectedly large number of positives, then we will have some undetermined samples as well. Yeah. So another nice property of this scheme is that the matrix is fairly sparse. So the pipetting time goes up, but it only goes up by a factor of about 2 or 3 or something. And that also means that the pool sizes go up compared to simple pooling, but they don't go up by too much. They are still in a region that we believe should remain viable. And because this is, there is so much redundancy here in the design, the same sample goes to multiple pools, there is some robustness. We can try to drop 1 or 2 of our tests and still try to reconstruct and see if there are some tests that being dropped lead to much better reconstructions. This would be a signal that those tests have some errors in them. And so this gives us some fault tolerance. We can tolerate a few experimental errors in pipetting, in cross-contamination, and so on. Okay. So we are now in clinical trials with a bunch of partners who are, Tata Memorial, so Malabar Cancer Center and NCBS and INSTEM, and we are initially focusing on tests that can tolerate a very large prevalence rate, because many of the samples that are arriving now, the rates of positives are very high. So this matrix that we have with 45 tests and for 105 samples can tolerate up to 20 positives in 105. It's designed for 10 positives in 105, but even if it goes up all the way to 20, we can list decode. We can give you, we can still give you savings. Okay. So what are our use cases? So this 105 and 45 would be one use case which might be suitable for testing labs that are seeing high prevalence rate. We would not deploy this maybe for symptomatics as written on this slide. We would probably advise them to take the symptomatics aside and pull the asymptomatics and then try to do this. And among asymptomatics, if you are still seeing 10% infection rate, which in fact happens to be the case at many places, then this would be an appropriate pooling test to run. The more aggressive, I think, pooling test should be run in the field, not for samples that are coming to you, but for samples that you are going and getting. Those are the situations where you would expect to have far lower prevalence rates. Those are the situations where you could expect to make a greater impact. So for example, you can think about hospital staff being tested every day. This would not be possible if there were a 2-round protocol, because it's going to take 8 hours. It's going to tie up your machines. It's going to tie up your lab personnel just testing the hospital staff every day. But now it's a single test. You do it for 4 hours, it's over, and you can possibly test hospital staff every day. You can possibly test CISF guards. You can test passengers at airports, police personnel. You can test journalists, delivery personnel, other high-risk population who come into contact with a lot of people, are in essential services, could potentially be super spreaders, maybe students who want to come back to campuses, maybe areas where people have been found positive and you want to, instead of quarantining the area, you just want to test everybody up and then see who should be quarantined in a sort of more focused way. So all these are possibilities. So yeah, you could test 400 samples with 63 tests, which might be more suitable for neighborhoods or large hospitals or passengers at the airport. This test can still be done by hand. So technician pipetting or 2 technicians pipetting can do this. So this test, the 1,000 samples and 100 tests, the amount of pipetting may become challenging for human technicians. So these are the ones that will benefit massively from liquid handling robots, which can do these kind of numbers in like 15-18 minutes. And in fact, we should really be thinking about deploying liquid handling robots in a big way across India at testing tests as big as this, where the savings are tenfold and you're still able to tolerate quite a few positives. Okay, so yeah, so what is the big picture here? The big picture is that there is this new technique. So pooling is necessary, because without pooling, we cannot scale testing. If we are doing pooling, then there is simple 2-round pooling, but there is an alternative, which is doing this kind of a non-adaptive group testing in a quantitative manner. So one thing maybe I didn't emphasize enough, and I should take the opportunity to emphasize now, is what is the role of doing it in a quantitative manner. The point is, suppose you have a test that comes out positive, and you know that one of the samples in the test is positive. If you are doing purely Boolean explanation for these tests, then you say the test is explained away. It has no more information for me. But if you are doing a quantitative explanation of the test, then you can subtract the viral load in the test from the viral load of the sample, which presumably you have estimated in some other way. And the residual viral load gives you an indication of whether there should be other samples in the pool that are positive or not. And both types of information, there aren't any or there are any, can be propagated through And that's what our reconstruction algorithms actually do. This is the kind of, they do it in a more global manner, not in this local manner, but essentially these are the kind of ideas. And so doing it in a quantitative manner can allow us to tolerate more positives. And that is what allows us to also be viable in regimes where, you have higher number of positives and so on. The problem with non-adaptive group testing always has been that it's more complicated. Who will know how to pipette this out? Who is going to sit and solve this problem? And so the solution that we have built for this is we have built an Android app called Vyom Smart Pooling. And this app guides technicians on which sample goes to which test. And it also, after doing the experiments, the technicians can enter their numbers into the app, and it solves for the viral loads on the cloud using the algorithms we have deployed, and it immediately returns to the technician all the answers. So we think that having this app can really increase the, so this can actually make this scheme practical, because the protocol now that the technician needs to follow is no more complicated than the protocol for individual testing. You just tell them what to pipette where, they get the result, they enter it into the app. Okay. All right. So this is joint work done with folks at IIT Bombay, NCBS. So Das at NCBS, Das at INSTEM has been one of the key figures in doing the experiments, and at the Wyss Institute, where Tom Schaus has done most of the experiments. Ajit is my colleague at IIT Bombay who works in compressed sensing. Sandeep and I have been thinking about the noise model, and the rest are graduate students and postdocs. I should say that Shop 101 has helped us build the phone app. So Aditya is one of the co-founders at Shop 101. Okay. So I have a few more slides, but I think I will rather give people a chance to ask questions, and based on that, we can decide what more slides I need to show. Yeah. So during the talk, were there any questions? I kept hearing some beeps. Yeah, so I think Uttar Pradesh claims they are the first, but I don't believe that. So before Uttar Pradesh was doing that, I think Kerala was already doing that.
I think, so Manoj, because I think the notification was issued on 14th of April, and that was based on
study ICMR to do you're absolutely
which slurs are applied to people who should know better. And hence, so there is like documentation I sent you, so that Uttar Pradesh was the first to start testing.
No, I've seen that. I don't believe
it, and they report everything how many they are doing. So that's and I mean I just responded like this because you said you don't believe it. So I actually posted the documentation to you, so you can read it. Thank you.
Sure. I think Prabhu had a question.
Yes, I just wanted to ask you a very nice work. Actually, what you said that up to 20% additional positives it can take over. But what if the number is suppose larger, like 70-80% infection? So under that,
don't do pooling. So in fact, it doesn't make sense to do pooling even for 20%. 20% is a boundary case where we do pooling because it might occur when we are doing it with 10%. We don't even want 20% to occur very often. Yeah. So if you are having that high degree of positives, then you should really be sorting out samples. The ones that you think are very high probability, you should be doing with individual tests. The ones that you think that you are doing just out of courtesy, you pool those, and then you see what you get. It will save you lots of tests. So you have to do some sorting before, if you are really in that 70% or even 30% bracket.
So your method is much more efficient than the binary type of thing, right? I mean, suppose instead of 100, the 50, 50, 25, 25.
So we have quantified. No. So the binary thing, that 50, 50, 25, 25 thing. The problem is that it is a deep method. Okay. Right. The problem with deep methods is that every round takes you 4 hours. So in the field, it is not practical. Nobody will do such a method. Yeah, if you have actually been in a biolab and you have done those kind of experiments, you will not do that binary search. you want it done, you want to move on to the next batch that is coming in. So this idea of latency is very important. And this is something that people do care about a lot in the field. It's not just, it's not just how few tests do I need, but it's also how fast can I get the test out of the door and go to the next batch.
And the other last question is, can you also give a weightage to each of these units? Maybe I missed this point, but what I mean is, giving a different quantity of say A, B, and C that you are going to put into one of the testing tubes. If you have that parameter, because right now you may be putting all the same quantity of A, B, and C samples. So instead of that, if you have a varied thing, do you think that will work, or that has been tried already?
I need to plug my laptop in. Give me a minute. My apologies. Just one minute. Yeah, that's not going to work either because we have a power cut and we are on generator. Okay, sorry. You asked, can I give a weightage to what?
What I mean is, right now you may be taking these A, B, C, 3 samples, same quantity. So instead of that, if you take a varied quantity,
we don't take the same quantity. We don't at all take the same quantity. In fact, we vary the ABC and we vary it in the way that we worst for us for the algorithm.
Okay, but this assumes that the viral load in all those 3 is the same. Is that correct, or?
No. We vary the viral load across all the positives. So we try to simulate as much as possible the real world situation. And in fact, when we don't know something about the real world, we assume the worst. We assume that the viral loads are as spread out as possible so that we have as little information about them.
Okay.
Yeah. So this is all done in an adversarial sort of setting for the experiments. We choose the viral loads that are hardest for our algorithm to reconstruct.
Manoj, how difficult is this to bring this 4-hour testing time down? Do you have a sense?
So there are other tests. So there is isothermal PCR. There are a bunch of other tests. So in principle, you might think that, pooling can be done with all kinds of tests. So the important thing is that the test needs to have good sensitivity. Yeah, hello?
Hi, Manoj.
Yeah, so I'm now, my laptop died on me. My apologies. So, so the important thing is, sorry, can you jog my memory? What?
Yeah, the question to you was, how can this 4-hour time be reduced?
So there are other tests that try to do things faster, and so people are working on a bunch of other tests. Now, whether pooling can be done in those tests is related to how sensitive those tests are, whether you can work with a large pool and still pick out molecules. Also, if the test becomes really fast, then you would not do a 1-round scheme. You would use the feedback. So then you would start doing binary search kind of schemes. The test is taking 5 minutes, then you don't mind running a 10 deep or even a 20 deep kind of scheme. So the thing to mention is right now the PCR test is slow, but it is wide. So the PCR machine usually has as many as 384 different wells, or 96 at least in the smaller ones. And so you are getting 96 tests in 4 hours. Yeah. So it has a good width. Now, how do you make best use of this width? If in those 96 tests, you could test 1,000 samples, then in 4 hours, you're getting 1,000 results, right? As opposed to 8 hours with 200 tests, you are getting 1,000 results.
and 200 tests really would already mean you need 2 PCR machines.
Can one expect to see pooled testing sometime in future in India? You think it's a viable thing, or it's uncertain? A month down, couple of months down.
Sorry, Sandeep, I can't hear you.
No, my question to you was: can one expect to see some kind of pool testing at workplaces, actively deployed in India in next month or couple of months?
I see. Yeah, so I would love to, see that happen. We are in clinical trials, and most of our time has gone into writing up the protocol and getting permissions and getting budget together. The actual experiments shouldn't take more than a week, and the analysis and all should be done pretty fast. So if things go to plan, then we could, we could then start pushing this to the regulating authorities and say that, you can consider doing this, and then based on how it does in the field, they can decide where to deploy this. And especially, workplaces, college campuses, airports are actually very good places for applying this scheme because it is a single round scheme. As opposed to simple.
You're the only group which is thinking about this. There's no other company worldwide which might do this.
So we are in talks with a couple of companies who are interested in doing this. So we see ourselves as facilitators. We will put our app out, we will offer these algorithms, and we will offer training in how to do this. And then people are sort of free to scale it to a large extent. Yeah.
Okay, so there are no more questions. Let's thank Manoj as well as Gautam for very nice talks. This was extremely informative, at least for me, and I'm sure for most people who attended. So I think that would be the end for today. I guess we'll regroup maybe in a couple of weeks. Thank you, everyone.
Thanks, Sandeep.
Thanks, Gautam. Thanks, Manoj.